Hello, this is Eric Topol for Medscape, Medicine the Machine, and I'm with my co-host, Abraham Verghese. And we're especially delighted today to welcome Walter Isaacson, one of the great biographers of our times, and his most recent book is Codebreaker, the Codebreaker, which is about CRISPR and genome editing. So, Walter, welcome.
Thank you so much, Eric. I'm a big fan of both of you, so it's an honor to be on your show.
Well, we are thrilled to have you. And I thought I'd set the stage because, the medical community, our Medscape audience, certainly knows a bit about CRISPR. It's the biggest life science impact, true breakthrough. You don't use that term lightly. I wanted to read two passages in your book that are striking to kind of set the stage, because they are really, it's kind of like AC and BC, where C is the CRISPR. And you captured it. You're a man of words, of course, but here, the first one I thought would be good to highlight. The issue is one of the most profound we humans have ever faced. For the first time in the evolution of life on this planet, a species has developed the capacity to edit its own genetic makeup. That offers the potential of wondrous benefits, including the elimination of many deadly diseases and debilitating abnormalities. And the other one, is actually way back in the intro of their book, which is also, I think, telling. And it says, there was a sense that we had crossed the threshold into a whole new age, perhaps a brave new world, like when Adam and Eve bit into the apple, or Prometheus snatched fire from the gods. So with that, these are pretty profound statements, Walter. Maybe you can just give us an overview, because you really delved into this. You're not a genomicist. You're not a hardcore scientist. You actually did CRISPR in the lab. You did a deep dive. So let's kind of get your overview sense.
I do think that we've seen certain revolutions in our time. In the early 20th century, we saw a physics revolution, much of which came out of the work of Einstein and others at the very beginning of this century. And likewise, in the second half of the century, we saw the computer, the microchip, and the internet sort of combine to create a digital revolution. And now we're on the verge of what I think is going to be a far more consequential revolution, one in the life sciences and biotechnology. It has many components, and it sort of begins to some extent with the sequencing of the human genome at the turn of this century, but also with genetic engineering, recombinant DNA. I think CRISPR takes us into an entire new league, because we can not only read the human genome and try to engineer things, we can actually make ever more precise edits to human, or for that matter, the DNA of any organism. And I think it's useful to have a broad discussion, first, of the wonders and beauty of nature. I mean, just out of curiosity, I think people should want to know this, but also because they'll probably have to wrestle over the next 10, 20 years with some of the implications of this. And by the way, it's just a beautifully exciting technology to understand how something works, especially when that something is ourselves. And I want to thank, you, but mainly all the graduate students in various labs in Berkeley and at the Broad and other places that went through the process with me of understanding exactly what the tracer RNA does and the guide RNAs do, and how you engineer a single guide, and all the things that I try to make exciting in this book.
Yeah.
Walter, I want to echo what Eric said. Just a tremendous book and a very compelling read. It almost read like a mystery. It just seemed to evolve on the page, which was wonderful. My question is, as a writer, as a biographer, when you approached this, you must have had an infinite number of choices as to how you would tell the story. And I'm just curious to hear how you arrived at the particular structure that you did use, and talk a bit about that, if you would.
Sure. That's a great question, Abraham, because I did spend a lot of time trying to sort through the process of what's the best way to tell a story. I have a natural proclivity to biographical narrative. I don't think it's anything new. I mean, that's how the Bible tells interesting stories and makes moral points. And It's got a great lead sentence, which is in the beginning, comma. So I tend to try to do chronological narratives with main characters that help bring it along. I do understand quite well that biographers, sometimes can distort history by making it seem like a guy or a gal goes into a garage or a garret and has a light bulb moment and innovation happens, when we know that it's a collaborative effort and a team sport. There have been a couple of books I've done in my life, including one called The Innovators, that was just a whole group of people without a main central character. And I thought of doing that, and there have been many good books. Kevin Davies has one, Editing Humanity. I think Hank Greeley has a great one called CRISPR People, that really bring you through all the people involved in this revolution. But as I went along and I spoke to a lot of people who could have been central characters, including George Church and Feng Zhang and Jennifer Doudna, and I flew to Berlin to spend time with Emmanuel Charpentier and Francisco Mujica, many people, Rodolphe Barangu, who were involved in this story. As my writing and thinking evolved, it seemed as a writer, a narrative writer, it's often useful to have a driving central character. And secondly, that Jennifer Doudna, doing it through her as the central character, had certain advantages, especially because she begins, her career, so to speak, in middle school when she reads a double helix and becomes absolutely fascinated with the notion that the structure of a molecule is a key and a clue to figuring out what it can do. She wants to become a scientist. Her guidance counselor says, no, girls don't do science. She's noticed Rosalind Franklin in the book, so she's inspired by such a character. And then, of course, as you all well know, she spends the 1990s under Jack Szostak and to some extent Tom Cech, doing the structure of various types of RNA, including self-replicating introns that answer the big questions, because Szostak had taught her, always ask the big question. And she says, what's the big question? And he says, how did life begin? And so by doing RNA that way, while a lot of the other scientists in the field, especially the men, were chasing the Human Genome Project and sequencing DNA, you had a lot of women, including Jennifer Doudna and Jillian Banfield and Emmanuel Charpentier, who are focusing on RNA, which turns out, at least in our time, to be a more interesting molecule. It actually does work. It becomes a messenger to tell ourselves which proteins to build, and hence we have the vaccines we now have. It becomes a guide that can take an enzyme and cut in a specified place, and hence we have CRISPR. So telling it through Jennifer offered the advantage of having a narrative that brings us through the history. Secondly, she then turns her attention to the ethical and moral issues, and along with David Baltimore and others who had led the Asilomar process in the 70s, she does a similar process to figure out what do we do with gene editing. And then, as you'll see, if anybody gets to the end of the book, she pivots to get groups of people in the Bay Area to take on coronavirus using both CRISPR and other forms of molecular biology. And so she became a very good narrative thread. I do hope, and I'm sorry for going on a bit on this, but it's sort of what a biographer has to wrestle with, that as you flip through the book, even just looking at the pictures and certainly reading the chapters, you'll see pictures of so many of the graduate students and the young investigators and the researchers. You'll see Virginia Sixnius and Martin Yannick and Prashant Mali and Jennifer Hamilton. And I wanted not only to get the stars of the CRISPR story to be main characters, meaning Feng Zhang, George Church, Emmanuel Charpentier, but all the principal investigators, because I wanted to show that creativity is a team sport when it comes to science.
Anyway, so what I want to, there's a couple of things I want to get to and give back to Abraham, but one of the things that I thought was remarkable that you did is bridge the book title, The Codebreaker, also is bridging to coding. That is all the things that you brought out with respect to the likeness of biotech here with CRISPR. And as you already mentioned, digital internet, the idea that people like Feng Zhang will move more to, is a symbol of the movement towards biotech. The Asilomar conference, which interestingly, whereas an AI conference was just held not long ago because of the dangers of AI, like with CRISPR. So I wonder if you could expand on that, as well as the point about that this was a basic science breakthrough that has been around for 1 billion plus years.
Right.
And, it's the way bacteria protect themselves against viruses. And it took basic scientists to do this. So can you just kind of put in perspective these two things? One, the kind of connection with the digital world and coding, and then the other, the importance or the essentiality of basic science.
Yeah, I do think that, during my youth and growing up, we all figured we had to learn how to code, and I knew C++ and Pascal and a few others. We made sure our kids learned how to code. I think it's going to be particularly important to have an appreciation for, not just C++, but C, T, G, and A, and the, letters of genetic coding. And the reason is because molecules have become the new microchip. I mean, whether it's with the mRNA vaccines or CRISPR, we were able to code molecules pretty quickly as soon as we see a sequence, say, of a spike protein of a coronavirus, and we decide we want to target it. So we have to get in that mindset that we're going to be recoding and re-engineering in order to create fast vaccines. Getting to your point about basic science, that's incredibly important to me. And I've written about it in The Innovators and in other books, which is the Vannevar-Bush concept of the linear progress of innovation, which is basic science tends to lead to discoveries, tends to lead to inventions and useful products that then help the economy. And Vannevar-Bush wrote that in Science, the Endless Frontier, I think in 1946. It became the model for the National Science Foundation, National Institutes of Health, and many other things. And in CRISPR, you see that quite a bit, which is the curiosity of Francisco Mojico and various other early CRISPR pioneers that are simply looking at a, more than 1 billion-year-old system that archaea and bacteria have created in order to, and they're puzzling over these repeated, clustered, repeated, interspaced sequences in the DNA of these microorganisms. And they're doing it out of pure curiosity. I mean, it's like Leonardo da Vinci, curiosity, pure curiosity. They're not trying to create a gene editing tool or anything, but that basic curiosity eventually leads to the understanding of the components of CRISPR, and then an understanding about how can we repurpose this bacterial system that's been around for more than 1 billion years and repurpose it to edit genes in ways that we target them, including in our own human species. That said, though, it's not a pure example of the linear model, and a lot of people I've read have questioned the linear model, and I think rightly so, which is it's more of an interactive dance between the applied scientists and the basic scientists, more of an interactive dance between the purely curious and those trying to do translational research, or for that matter, business people. Whether it was, the linear model that Vannevar-Bush talks about is people studying the conduct of electrons on the surface of semiconducting materials and figuring out the quantum mechanics of it, do that out of just pure basic research, but we end up with the transistor and the microchip. Well, yes and no. We end up with the transistor at Bell Labs because Bardeen and Shockley and some of the theoretical physicists are sitting there with experimentalists like Walter Brattain, but even business people are saying we got to amplify a phone signal between the two coasts, or we've got to, pole climbers with grease under the fingernails who know how amplification works. And that interaction creates a transistor. Likewise, with CRISPR, you have Horvath and Barangu working at a yogurt company or yogurt and cheese company and Danisco. And they are looking at the sequences that are just, of bacterial cultures that are getting destroyed. And they're noticing from the historical record of these cultures that the viruses that have attacked them, a mugshot of those viruses has been put in the clustered repeated sequences. So I think even in CRISPR, you see this beautiful dance between basic curiosity-driven research and applied and translational research.
I think one of the fascinating side stories of the book is the necessity or the trend towards all these scientists making links with the business world, patenting the technology, starting companies. I'm puzzled that, for example, Stanford, for example, embraced the idea of commercializing and capitalizing on these discoveries, where Berkeley was initially reluctant. And now it seems like everybody's starting a company while in academia. Pretty much everybody embraces it. Can you talk a little bit about that dance and how it's evolved and where you see it going?
Yeah, I think that's important. I do it both in this book and Innovators and Others. And you're sitting there at Stanford as we speak, Abraham. And you know that in the 1970s, in particular, under, I think it was Fred Terman, who was then provost or dean of engineering at Stanford, people who were like Hewlett and Packard coming up with great ideas and intellectual property out of Stanford were encouraged at Stanford to commercialize them, to get patents. And this happens even in the 70s, not only at Stanford, but with recombinant DNA and Peter Boyer and the people who started Genentech. So when you look at the valley in which you're situated, Abraham, you have companies that started it off, such as Hewlett Packard and Genentech and others that come out of Stanford and the other universities there. You on the East Coast, you have Harvard and MIT, which had up until then been at the forefront of digital technology, of creating the routers for the internet and everything else. But there's a condescending attitude towards commercializing content at all sorts of traditional universities. So Stanford helps get a leg up and they become amazingly successful, whether it's the recombinant DNA patents or I assume Hewlett and Packard, and for that matter, Google spins out of two, grad students, Larry Page and Sergey Brin at Stanford, and they're encouraged not to finish their doctoral dissertation, but to get out there and find a garage somewhere and create a company. So now I'm at Tulane University. We're in the third wave, meaning we've watched Harvard and MIT and others catch up, and so many other universities, University of Washington, so many catch up with the Stanford approach. And now, whether it be University of Texas or Vanderbilt or Tulane, we're trying to create these centers that will have patent attorneys, that'll have venture capitalists, that'll have innovation centers. Now, that can go a bit too far. And in my book, The War Between Berkeley and the MIT Harvard folks over the CRISPR patents, I think gets a bit unseemly at times. It reminds me of the story of Jack Kilby and Bob Noyce at Texas Instruments and Intel fighting over the microchip. And finally, Noyce calls up Kilby and says, let's quit fighting over divvying up the proceeds so we finish robbing the stagecoach. And they just cross-license their patents. I kind of hope the Broad and Berkeley would do that. But patents, to me, are an important part of the process. They drive people, just like competition for prizes, drive people to stay up late at night and to work on weekends and to have the funding in order to do things. So there's a great backlash against patents, especially in the era of COVID vaccines. But even with my daughter, who is of the generation who's not as much in favor of intellectual property, I defend the desire to commercialize and patent things as long as those patents are used the way Bell Lab used its patent on the transistor to help spark an industry rather than suppress an industry.
Medicine and the Machine will be right back.
Welcome to Medscape's latest podcast series, Medicine and the Machine, featuring Medscape's editor-in-chief, Dr. Eric Topol, and master storyteller and clinician, Dr. Abraham Verghese. Remember, you can find the latest in medical news, expert perspectives, clinical tools, continuing medical education, and more at medscape.com.
Well, one of the extraordinary parts of this to me, because, I knew of Jennifer Doudna's work, but of course you traced it from her sixth grade reading of Watson's book to all the way through to the Nobel Prize. And what I was struck by, Walter, one is how you got this remarkable access to her, because she had to confide in you and really spent a lot of time with you to bring you along. But she was brought along from a chemist. I mean, I guess she almost was a French major, which is kind of amazing, but she a chemist who then, does this extraordinary work in RNA. And then with Emmanuel Charpentier with the cells, showing how CRISPR works, certainly not in human cells, which is a bit of a gap. But then she's obviously very competitive, which is a good thing. And then she, basically expands from, now she's getting into human, diseases, human trials, as you already, Abraham, and you touched on, companies and ethics and public policy. I mean, that's from a chemist to do that all the way to, the big picture of, the universe is pretty transformational. Can you comment about that?
Yeah, I think that it's important for scientists to do what you do, Eric, which is also be public policy intellectuals, also engage in education. And I think that there's been a problem with science really over the past century and a half, which is when I wrote about Benjamin Franklin, he loved science, and he would have thought you were a Philistine if you were not involved in science, if you didn't know the difference between even an integral and a differential equation, or if you didn't understand gravity or the Gulf Stream or botany, and same with Thomas Jefferson. But then Einstein comes along, and science becomes a bit intimidating. People think you have to be an Einstein in order to understand it, and it becomes slightly frightening. I mean, we get, a picture of Einstein on the cover of Time magazine with a nuclear a bomb cloud behind him. And so when I was growing up, we were living in C.P. Snow's two cultures, where those who were humanists, kind of would be unashamed to say, I don't understand science, I can't do math. And sometimes scientists were a priesthood in which they would, when I went to college, I loved science, and I studied biology a little bit, and I studied physics. But as somebody who wasn't majoring in the hard sciences, you didn't really feel all that welcome going to physics and biology classes. Whereas if you were, majoring in molecular and cell biology, and you took a Shakespeare course, you'd feel perfectly welcome. So I think scientists are sometimes formed into a priesthood where they don't popularize, or even they look down upon popularizing what they do. Well, this leaves a role for a Jennifer Doudna to become a public policy intellectual, the way a David Baltimore did, the way you have, Eric. It also leaves room for people like myself who are not bench scientists to say, let me hang around the bench and teach me what you're doing so I can translate it into something that the public will not only understand, but be able to discuss, appreciate, see the beauty in.
One of the most exciting parts of the book, I think, because most of us were following that dramatic episode,
right,
was the Hong Kong conference, where the Chinese scientist reluctantly presents his data on editing the gene of these babies. And talk a little bit about that. And also, are we entirely safeguarded from that sort of thing based on the guidelines that Baltimore and all of them have set up? What are your fears about CRISPR technology going forward?
Yeah, well, my fears, first of all, are overwhelmed by my hopes for it. I was actually just looking for it. I get every morning I wake up, and I got one this morning, which I was thinking of showing, but of people send me a picture of their daughter, or in this case, a son that has, I can't remember the name of the monogenetic situation, saying, can you put me in touch with the people in your book? Because they say my son's going to die in 3 years and he can't walk, or whatever. And so my heart goes out and I keep saying, all right, let's make sure before we tap on the brakes too fast here, we understand the promise of this, especially for horrible genetic conditions, including common ones like cystic fibrosis, muscular dystrophy, et cetera. Will people start designing babies? I kind of go through the case studies in the book that I worry about. And certainly, I think we look at what He Zhuangqi did in Hong Kong, what he announced, and we were all appalled by it. He made inheritable germline edits in early stage embryos. But when you look at what he did it to knock out a receptor for, a virus, HIV, the virus that causes AIDS. And coming out of the pandemic, there might be people who say, well, remind me again why it would be bad if we learn how to do it 20 years from now very safely to reduce the number of receptors for viruses and study the unintended consequences and maybe find a way to fight viruses. So I think the research should progress. I do worry about rogue scientists. In the book, Jennifer Doudna describes a nightmare in which somebody says, I want to learn about your technology. She goes in the room, person looks up, it's Adolf Hitler. So yeah, we can have the nightmare that state-sponsored eugenics, and the Russians may use it for super soldiers, whatever. I think it's more likely that we're going to have a free market libertarian eugenics, meaning if we just allow parents to buy 20, 30, 40 years from now, better genes for their children, and the rich can afford to make their kids, say, more muscular or have better blood cells that carry more oxygen, or maybe even better memory or certain synapses, processing power. These are well into the future, but a system in which the rich get to buy better genes for their kids and in which we start editing out the diversity of the species. One of the more interesting bioethicists in my book is a 17-year-old kid named David Sanchez, and he loves playing basketball, except for when he doubles over in pain in the center of the court because he has sickle cell. And the people that, both of you all know, in fact, I think it's at Stanford, he's being treated, Abraham, they tell him, well, we may be able someday to edit your sperm cells or reproductive cells so your children won't have the sickle cell defect, if you want to call it a defect. And David says, that's great. That would be wonderful. And then a little bit later, he says, well, maybe that should be up to my kid after my kid is born. And you ask, well, why do you want your kid to have sickle cell? He says, well, no, but sickle cell taught me a lot. It taught me persistence. It forged my character. It taught me how to get up off the floor when I fell down. And maybe we shouldn't edit that out without the permission of the kid. And then later I asked him, he said, no, I've decided I want my kids not to have sickle cell. I said, what about persistence? I said, well, I try to teach them empathy and persistence, but I don't want them to feel the pain. I use this example not to say there's a right answer, but that we all should have first thoughts, second thoughts, third thoughts about what type of things do we design in and out of our species.
Yeah, well, the point you're making regarding somatic versus germline editing are crucial, and you develop that fully in the book. And just to close the chapter about the CRISPR babies, actually, I didn't even realize that until I read, Walter, how he was convicted and is in prison for 3 years with a very large fine. That was really quite interesting. I knew he was in trouble in China, but I didn't know he got into that much trouble.
Right.
But, the somatic thing is really hot right now. Undoubtedly you saw, and in fact, you gave us a preview. Just my friend Seth Katharison and his Verve text treatment that just reported in a non-human primate the ability one shot to knock out the PCSK9 editing and get low levels of cholesterol, LDL cholesterol, down to what you would want for a lifetime to prevent coronary disease, no less the effects with David Liu's base editing on progeria.
Mm-hmm.
And then just yesterday in Nature, sickle cell, one shot base editing. And this is, of course, in mice, not just in people. But this is accelerating. The somatic mutations, as you develop for Huntington disease, sickle cell, and then possibly other monogenic conditions, has extraordinary excitement that we can see in the short term. I mean, we're already seeing it. And I think that's what's so important now, is not to get hung up in the germline, because that's where it gets dicey. Would you agree to that?
Yeah. I mean, I think it's just extraordinarily important that people understand the promise of this, as well as perhaps the potential perils down the road, and that we're not near germline edits that could be widespread. I mean, even though Verghese did that in China, we can go very slow on germline edits. And in the meantime, we should celebrate what David Liu is now doing at Harvard with base and prime editing, and all the ways that congenital blindness, and as you said, blood diseases, cholesterol, these things are going to be great boons to society. I did want to start the conversation, though, and that's why I do have, let's go step by step of what inheritable editing we would do, because I think 10 to 20 years from now, we're going to have a consensus on, whether it's like, trafficking, sex trafficking or shoplifting or anything. We have a consensus on what we're going to allow and what we're not going to allow. I think we ought to start having that discussion. And I can see reasons why we would want to do germline editing at some point.
Mm-hmm. Right.
For example, the sickle cell, Victoria Gray was the first, I think, to get cured of sickle cell down in, she's from Mississippi. And that was last year. But that cost more than $1 million. I mean, it's a stem cell extraction and transplant, then the editing in order to do that. If a David Sanchez, we can edit so that we knock sickle cell out of the human species, that's going to be affordable rather than, and when the Senate held a hearing on this after Verghese did this thing in China, Jennifer Doudna, George Church, others were there, and they were waiting for the senators to want to ban, CRISPR editing like that. And the talk turned instead to, won't this be a way that it'll be cheaper, so we're not going to break the bank every time we have anything from, as you put it, cholesterol problems to sickle cell. So I think we have to go carefully down what is a slippery slope, but if you do it step by step, hand in hand, it becomes less slippery.
Walter, I think you did a wonderful job of conveying the excitement of science, the adventure. And, I walk down these corridors in Stanford and I always wonder just, exactly what's going on in there. And most of that, no one ever hears about till they publish. And even then, if you don't understand the jargon, you miss it. So I loved all the compelling stories about individual scientists, but I confess the one that I loved the most and felt I would have loved to continue to hear more of is George Church. He sounds like a fascinating sort of Renaissance man in the midst of, hardcore scientists. And talk a bit about some of your favorite characters in this story.
Well, George is certainly one of them, and he's a main character in the story. There's a book, Regenesis, about him, and he's trying to resurrect the woolly mammoth using gene editing, and he's been in the gene editing trade for a long time. I mean, it's not just CRISPR. He goes back to talons and ZFNs and others, and he looks a bit like the woolly mammoth, which maybe there's a sense of kinship. He wants to resurrect him with his bushy beard and mild halo of hair, and he's very provocative. I mean, he's the one in the book who, when I say we don't want the rich to be able to buy better genes for their kids, he said, what's wrong with some rich people making their kids smarter? He's provocative in that way. And yeah, I think he could have been a central character of the book, and he has been central characters in other people's books. He is very close to Jennifer. In fact, when there were certain prize given out and George was not included in it, she invited George to be her guest and donated the prize money to the foundation George has started with his wife, Ting, called it's about educating people about genetics. And another character that's particularly interesting to me, and I just had drinks with him in New Orleans a few days ago, is Josiah Zayner. You may remember him. He's the biohacker, the one who in his garage with the Odin is doing his own CRISPR things, making his own DNA vaccines, injecting himself with a CRISPR treatment that will regulate myostatin so he can have bigger muscles. And I remember going back, Abraham, talking about Stanford area. You know, I'm old enough to remember the Homebrew Computer Club. And when the hackers and hobbyists took over the use of microchips from the big corporations and created, you know, Steve Jobs and Wozniak and Bill Gates, the first personal computers, how hackers and hobbyists and phone freakers were at the forefront of democratizing the digital revolution. Well, that's not easy to do in the biotech revolution, because if you're sitting in your garage, as Josiah Zayner does, concocting CRISPR and vaccines and other things, it's a little bit more dangerous than even hacking into the Bell system to make free phone calls. But there is something good about democratizing science. And with all due respect to the two of you, pulling it out of the castles of, you know, Scripps and Stanford and other places, and saying you too can, you know, just like I grew up here in New Orleans, my father was an electrical engineer. I grew up in, you know, our basement soldering circuits. I felt I could have hands on. So I like Josiah Zayner, and he's going to be sort of the ethical issue, because when we ban CRISPR for certain things, then people like Josiah Zayner are going to be selling it on the internet out of their garages. So he pops up just like Puck does in A Midsummer Night's Dream every now and then to say, what fools these mortals be. And he's sort of the provocative character who's the wise fool. And so I like him and George Church a lot. Well, and of course, I fell in love with Emmanuel Charpentier, but that's a different subject.
I do want to get into that subject. But before that, that was one of the things that struck me, Walter, about your balance, because, you know, you're coming in favor of Zayner and democratizing CRISPR, I thought was really very interesting, along with your forward-thinking view of where germline editing will go over time. But to me, the most heartwarming part of a book, which is riddled with conflicts between people, like, you know, the tension between Feng Zhang and Jennifer, which, you know, in many ways, George Church was the person who was most supportive of Jennifer. But even between George Church and Feng Zhang, there was conflict because of his somewhat betrayal or lack of real loyalty and communication. But to me, you're bringing together Jennifer and Emmanuel, who were about to get the Nobel Prize together, but they were at odds. And you went on a mission to get them to rejuvenate or renew their friendship. Can you tell us about that? Because I thought that was masterful.
Well, it was not fully intended, but it just evolved that way. And like the Heisenberg principle says, sometimes we observers affect the things that we observe. And I love the relationship in the science and intellectual relationship between Jennifer Doudna and Emmanuel Charpentier. I spent time in Berlin in Emmanuel Charpentier's lab, and she's a very guarded but genuinely wonderful person, but with a shield around her, Emmanuel. And as you said earlier in this show, Jennifer she was going to be a French major. So she always she loved Emmanuel Charpentier, and they had this great bonding when they were working together. And then they drift apart. And it reminded me that these are human stories, just like the story of Steve Jobs and Bill Gates is a human story where people compete, they collaborate, they drift apart for reasons neither side quite knows. And Jennifer is, as you know, because you know her well, Eric, a very sincere person and open and self-aware. And she would ask me, when you talk to Emmanuel, tell her I'd love to get together with her, but we seem to have drifted apart. Is there any, is she annoyed at me? And yeah, there were some annoyances, like the Kraken creation book in the first person that Jennifer did with Sam Sternberg. Emmanuel said, well, that she was thrusting herself out too much. People get a little jealous or a little rivalrous, but there was no reason they should have fallen apart. And there was a CRISPR conference coming up at Cold Spring Harbor last year, which was a virtual conference, of course, last year. And I had just come back from Berlin, and Jennifer Doudna said to me, tell me what can I do to try to restore my friendship with Emmanuel? I said, well, why don't you invite her to present at this CRISPR conference? And she said, well, that's a good idea. And Jennifer talks to Maria Jansen and others and invites Emmanuel. And I urge Emmanuel to do it. And she says, no, I'm too busy, which seemed somewhat odd. It's a 3-day conference on Zoom, most of us could, but it was clear she didn't quite want to do it. So I was puzzled. I said, why? And finally I said, well, how about just you and me and Jennifer right after the conference ends on Sunday? Why don't the three of us just get together on Zoom? I need it for my book, I said. The book begins, the scientific journey begins with you and Emmanuel and Jennifer walking the cobblestone streets of Puerto Rico. I said, I need to have some closure here. Why don't I have a joint interview about what your relationship was? And surprisingly, Emmanuel said, I would love that. That would be really important to me. And so Jennifer said, I would love it. So I had all these questions. It was a Zoom screen like we three are on right now, even though your listeners can't see it. And like any Zoom screen, I'm there. I've got a list of questions. I'm going to start guiding the conversation. But the minute they both get on Zoom, they start asking each other, how's your son, Andy, or what's happening here? And so I just turned off my video. So it was the two of them together, and they talked for 45 minutes. And to me, it wasn't a necessary part of the book, but it did talk about these are human endeavors.
Yeah, I thought, you're not just write a book, but to bring these two phenoms together. I thought that was a great contribution. I mean, just apart from everything else.
Well, then the Nobel, let's admit it, that the Swedish Academy did more to bring them together about a month or two later.
Well, but it might have been with some cold stuff still. You got them back. That was pretty clear.
And for those of you who are in the med field, as you know, the Cold Spring Harbor conference this year is in late August, and it is virtual again. And the keynote speaker is Emmanuel Charpentier with Jennifer Doudna. So all is right with the world. Tune in.
Yeah, it's fantastic. Yeah. Well, I think we want to let you go, but I guess, before we wrap up, I am an avid reader, but I don't remember a book that had as much impact. Of course, the field is one that I followed closely, but the way you told the stories, the way you brought out all the characters, the human side, no less the science side. It's really quite extraordinary. I hope that our Medscape audience gets into this book because they will appreciate the importance of this discovery. It isn't just a prize-winning thing. It's something that, like I said in the outset, AC, BC, where C is CRISPR, and the impact here will be probably the most profound thing, certainly in my lifetime, Walter. I can't imagine, in terms of life science, there will be a breakthrough like this for some time to come. I don't know your thoughts about trying to think way out in the future about that.
Well, the Nobel Committee, I set my alarm in New Orleans to 4 a.m. so I could watch the live stream on the off chance that CRISPR would win the Nobel Prize this past October. And when the Nobel Prize secretary began reading and said, not only for a discovery, but it brings science into a whole new epoch, brings science into a whole new era, I realized it was going to be CRISPR, because that's what's happened. As you say, it's before CRISPR and after CRISPR. And what I wanted to do, and I appreciate you and Abraham, what you do, is to show ordinary people what happens in labs like yours and what happens at research places like yours. I don't think people get how the scientific method works these days. And that's so important in different fields. But also, what do people do in a lab? How are they driven by curiosity? And by the way, how are they driven by patents and prizes and priority of publication? And I admire so much what people in the medical research community are doing, that having the small chance to make it more understandable to a wider audience was not only an honor for me, but it was a joy.
Walter, it's just a wonderful contribution, and your great skill is to tell the story in such a compelling way and connect all these dots. So thank you so much for giving us your time and allowing us to share the experience of this wonderful, wonderful book.
Thank you.
Thank you.